Hey, Aaron. Good morning.
Hi. Hi. How are you?
Good, are you?
It's great to see the HKUST campus
Oh,
background. I miss it.
thanks. So how's my voice? It's good?
Good. Yeah, yeah. Do you want to test your slide?
Yeah, sure. Let me share my screen. Is it good?
Yep, looks good. Hi, Chih-Ying.
Hi, Aaron. How are you?
Nice to meet
Actually,
you.
nice meeting you here.
Hi, June. I don't know whether you remember. June, I overlapped with you at Hopkins for a little bit. Yes.
Yeah,
You
yeah.
are,
Your
yeah.
name actually, when I saw your name, I kind of think that we know each other before.
Yes.
Chih-Ying, do you want to test your slides?
Yes.
Jin, maybe you could stop sharing.
Oh, okay.
Does it look good?
Yeah, it's great.
Yeah, good. I forgot. Who was your supervisor in Johns Hopkins?
King Waya.
Oh, you're Ching Su, PhD student. I see. Okay.
Yeah. Yeah.
Yeah, I was with Rick at the time. So we're on the same floor actually.
Yes. Yeah. So thank you so much, Aaron, for the opportunity. Yeah, that was
Oh, yeah.
pleasantly
Yeah.
surprised when I saw that I'll be getting the talk with June. Yes.
Oh, yeah, yeah. Yeah, it'll be good. And then I'll see you in Ventura for the GRC.
Yes. Yes.
Are you coming to the wine tasting?
Yes. Yeah. I plan to drive up on Sunday. Yes.
Okay, yeah, try to get there as early as possible because it'll be really amazing wines.
So what time will you start?
Oh, it's later. It's after the session actually, so that doesn't matter. But it'll just be
Oh,
good to get there early anyway.
yeah.
Just for
So are you back in California?
yeah, I was in China for a year, and then I've just been traveling all around. I was in Ireland last week and stuff, and now I'm in California for a little bit.
Aaron,
Hey, it's
Hi.
along.
hi. Hi, Jing. Hi,
Hi.
Jing. Nice
Hi,
to see you.
Hey
Hi.
there.
see you. Hey, John.
How's it
I
Hi,
going? Good
John.
to see you, Jen.
I love
Hope you're
that
doing well.
background, the HKUST background.
Mm.
Oh, thanks.
Maybe
You
it's not a
miss it.
background. Maybe it's real.
Yeah.
Oh,
Oh,
it's a
yeah.
background. It's background. I can see this from my window though, but you won't be able to see me if I face my window.
We were supposed to have the GRC at HKUST, but they switched it to Ventura.
Yeah, the COVID disrupted everything.
Yeah. But we'll come back. We'll to HKUST someday.
So, Zilong, how's the situation in Shanghai? Can you move around
Yeah,
freely?
yeah. Largely come back. We come back to work. Had some dying out sometimes, but not always. But yeah,
That's
coming
good.
back.
So there's no more restriction.
Yeah, not much, not much. So at the beginning, in the middle of June, there's no dining out. But now it's like in the beginning of July, it started to dining out. It's just still some small location has been locked down for 1 week or two, but
How
most of the
often do you have
rest
to
restaurant.
How often do you have to take a COVID test, Zong?
That's a good question. That's a good question. So I do that every other day. So we have to come to the campus. We have to do the 72-hour COVID test within a
You do it
72
every other
hours.
day?
Yeah, so I rather to do it every other day in case I forgot sometimes. So the easy thing is we have some local tests. Now they have a small like shelter thing in every community, every apartment, even in several in the campus. So it's very convenient to do that. Yeah.
Now that I'm out of China, I realize how stupid it is. Just if you go to like Europe, everywhere, it's like, I don't know, it's just pretty stupid what China's doing.
It's already everywhere. Yeah.
Yeah. It made sense when we were there, or it didn't make sense, but we didn't know any better. But now that I'm out, it makes it all the more silly.
Hopefully we can get over this. So
Well, that's the problem. I think everyone in China will get infected eventually. It's just they're just delaying it. It's going to take 6 or 7 years. They don't realize it yet.
oh no.
I ran into Yang Dan in the airport yesterday.
Ah.
Hi, Jin.
Aaron's meeting everyone everywhere.
Hey, Jen, good to see you, Jen.
Good to see you, John.
Jin's not coming to our GRC, unfortunately.
She's not.
No,
She was invited though, right? You were invited. I thought I saw you on the list.
She said that there's some other more important meeting for her to go to.
Not possible.
I know.
Oh,
Oh, that's too bad. I was hoping to see
Sorry.
you.
yeah. Sorry, miss this meeting because Aaron never told me that he switched the meeting. When he switched the meeting, he switched to this day.
Well, that's Aaron's fault then.
Is that right, Aaron?
That's
You didn't-
Aaron's fault. Aaron screwed up.
Okay.
I'll have to mark the meeting lower when they give us the survey.
Okay. Ming Jia is the chair next time, so he'll deal with it.
Well, definitely looking forward to all of you coming to SFN. November for sure.
Right.
Let's have a party at your house.
Yeah, well, you've been saying that. I have been saying yes.
You're just hoping he'll
Diego.
forget. You're hoping he'll forget, right?
Yeah, I'm
He's not
hoping
gonna forget.
that's right.
DeLong, are you going to SFN?
Sadly, no. In San Diego, I miss San Diego so much.
Well, you never
Wow.
know if everything got opened up by October. So every month can change, right?
But how was I forgot the SFN last year? Was it Chicago? It was virtual, right? Last year
No, it was
wasn't
so-
in person. Yeah. Yeah, so it's supposed to be the huge meeting. Remember?
how's DC, John? Weather-wise, life-wise?
I was back there last month. It was hot and humid.
Mhm.
Summer in DC is pretty brutal.
Yeah,
Winter too.
not really. Winter is pretty mild, DC, right? Unexpected.
Yeah, the weather has gotten kind of extreme.
Yeah, Boston winter is definitely harsh.
Aaron, you get started?
Okay.
Yeah, sure.
Okay, welcome everyone to another week of NeuroZoom. This next Monday, we won't have a session because there's a neurobiology GRC that most of us will be attending in person, but we'll be back at it the following week. So no NeuroZoom next week. 2 great talks today. Zilong. Oh, and then please let Zilong and I know if you want to talk about your research. We have spots available for September and beyond. So keep tuning in. We'll keep bringing cutting edge science to you. Zilong, do you want to introduce Jun?
Sure, great. So it's my great pleasure to introduce Jun Xia. So it's a great colleague of mine. So Jun has got his a medical degree. Actually, Jun has trained as a physician in Shanghai Medical School, which is one of the best medical school in China. So after training physician, he went to actually the old institute of mine, so Shanghai Brain Institute in Chinese Academy, where the traditional campus we were all are in. So after I believe the short master degree, so Jun went to Johns Hopkins. From pursuing his PhD with Rick Hunar, where he learned all the right in the exciting age of the synaptic protein discovery. So in 2001, Jun got his PhD from Johns Hopkins. So right after graduation, I believe he was recruited by Nancy Ye to the Hong Kong University, Hong Kong UST ever since. So Jun has started an amazing career in Hong Kong. Have made a number of discovery to identify a series of interesting partner synaptic proteins. So Jun now also has a position in Hong Kong UST, has a side campus in Shenzhen. So today, Jun will talk about the role of neuroligin 2 associated proteins in inhibitory neuron synapse formation. So welcome, Jun.
Thanks, Zilong, for the nice introductions. And it's my great pleasure to share some of our recent work with you. Okay, let me share my screen first. And also, it's great to see some of the old and new friends online. Okay, it's not easy nowadays to travel outside Hong Kong. But can you see my screen?
Sure. You want to go to slideshow mode?
Good. I just put on the slideshow. Okay, so basically, I've been interested in synapse function and also synapse formation for a while, and we all know that synapse is one of the key connection point for neural network and important for neural integration and network formation, which I don't want to spend too much time on this. So there's a lot of key molecules have been identified that are important for synapse formations. One of them is the neurexin and the neuroligin pairs that had been identified many years ago. And we also know that mutation of neurexin neuroligin could contribute to a number of psychiatric disease, including autism. So what's interesting, what we found interesting a few years back is that neuroligin, which has a number of isoforms, they have distinct role in inhibitory and also excitatory synapse formation. So neuroligin 1 is mainly responsible for induced formation of the post-synapse structures, while neuroligin 2 mainly induce the formation of inhibitory synapses. But when you compare the sequence of neuroligin 1 and neuroligin 2, they're actually very similar. So the major difference actually lies in the C-terminal regions. So we saw that one way to figure out what actually mediates the assembly of excited synapse and inhibited synapse could be helped by decipher what actually associated with the neuroligin 1 and the neuroligin 2 proteins. So basically, the strategy we use to do is rather simple. So we generate antibody against both neuroligin 1 and neuroligin 2, and we use those antibody to immunoprecipitate the protein associated with them. And in this case, today we focus on the neuroligin 2 binding proteins. So the strategy is rather simple, as I mentioned, that basically we pull down everything associated with neuroligin 2, and then we use mass spec to identify what comes down with it. So this is actually it's not hard to find protein, but the hard part is to find the protein that specifically binds to neuroligin 2. So in order to exclude the non-specific binding partners, so what we did is that we use the neuroligin 2 knockout mice as a control. So basically we look at what actually the protein comes down from the wild-type mice, and then we look at what comes down from knockout mice. So whatever that comes down from both will be excluded. So we only look at the protein that specifically comes down from the wild-type mice. And even that, we still get a lot of protein that come down from the wild-type mice, and we repeated it multiple times. And eventually, we narrow down about a dozen proteins that can specifically pull down from the wild-type mice, but we did not find anything come down from the knockout mice. And one of the protein I want to share with you today is a protein called LHFPL4, which we'll call L4 in the subsequent talk. So L4 is a protein that initially identified from lipoma. And the function of L4 at that time, when we first identified, wasn't very clear. It's a transmembrane protein. It's actually a small protein, only 200 some amino acids, but with 4 transmembrane domains. And when we identified, we also did a quantitative IP to measure how much of L4 that associated with neuroligin. And what we did is that we immunoprecipitate neuroligin 2. Then we look at how much after we do a consecutive co-IP until we completely deplete neuroligin 2. Then we look at how much L4 is still remain. And by this way, we can quantify how much neuroligin actually associated with L4. And in the end, what our quantification shows that about 1/3 of L4 in the brain that are associated with neuroligin 2. And as we know that one protein usually associated with hundreds of other proteins. So with 1/3 of L4 associated with neuroligin 2, actually it's the most redundant protein we found that could associate with neuroligin 2 in the brain. So we conclude that L4 is a major binding partner of neuroligin 2. And then we went on to look at the localization, which is not surprisingly that we found that L4 is specifically enriched at inhibitory synapse, and it co-localizes quite well with neuroligin itself, which is not surprising. It also co-localizes well with Jeffrin, an inhibitory synapse marker, as well as gamma-2, which is inhibitory neurotransmitter subunit. And when we look at the PS75, which is excitatory synapse, we do not see very good co-localization. And this shows the quantification data shows that L4 is highly co-localized with inhibitory synapse marker, but almost no co-localization with excitatory synapse marker, indicating that L4 is rather specific for the inhibitory synapse. And we also look at the localization of L4 under super-resolution, try to determine whether L4 is localized at pre-synapse terminal or post-synapse terminal, because we know that neuroligin by associated with neurexin, it can actually pull down some pre-synapse proteins. So what we do this is we use a pre-synapse marker, which is GAT65, and also inhibitors post-synapse marker Jeffrin as a marker to tell whether this protein is pre or post-synaptic localization. So as you can see that this is L4 green with Jeffrin, which you can see that it's very hard to distinguish L4 from Jeffrin under super-resolution. But however, when we do double staining of L4 with the GAT65, you can see that you can clearly separate L4 from GAT65. And this is indicating that L4 is actually co-localized with Jeffrin at the post-synaptic site, but not on the pre-synaptic site. Then we went on to look at what could be the function of L4 by knocking it down. And the knockdown experiment shows quite clearly that we use 2 different SHRI. When we knock down L4, that we see a depletion of both Jeffrin, which is a post-synaptic marker, as well as VGAT, which marks a pre-synapse inhibitor synapse. And we can see here that both 2 SHRI will be able to knock down the VGAT quite nicely, but control siRNA doesn't do this. And when we look at the excited synapse marker, in this case, PS95, and also V group 1, that we did not see any changes. When we knock down the L4, the PS95 and V group at synapse is just as usual. And this is quantification data shows there's no change in V group 1 and PS95 positive clusters. So indicating that L4 knockdown will only affect the inhibitory synapse formation, but not excited synapse formation. And then we generate L4 knockout mice. And what we found is that the behavior, the heterozygous mice doesn't seem to have a serious behavior problem, but the homozygous mice had serious motor behavior problem. As you can see here, that it takes much longer time to cross beam to reach the other end of the terminals. And in addition to the motor behavior, we also find that when we use PTZ, which is a GABA receptor antagonist, to induce seizure in the mice, and we found that the homozygous mice is much more likely to have seizure compared to the heterozygous or the wild type mice. So this actually is expected when the mice, they have, when we knocked out L4, the inhibitory synapse was actually disrupted in those mice, and they actually have a higher number of excited synapse, and they're more likely to have seizure in those mice. So based on those results, that we propose that L4 actually will be able to associate with neuroligin 2, then will further link with GABA receptors. In the meantime, when work was ongoing, Susumi group actually they use GABA receptors and did immunoprecipitation and identified L4 as GABA receptor binding partner as well. So L4 could serve as a link between neuroligin 2 and the GABA receptor, and also bring GABA receptor to the inhibitory synapse. And when we knock out or deplete L4, this will lead to the disruption of the inhibitory synapse formation, and will lead to motor behavior deficit in those mice. So this is one of the protein we identified. And in the remaining time, I will share with you another protein that we identified from our study, which is which is a ubiquitin E3 ligase, which was also identified as neuroligin 2 binding proteins. And each has three main domains, which is C2 domain and HEC domain, and also 4WW domain in the middle. It belongs to a family called the NEDD4 ubiquitin E3 ligase, and there's a number of family members in these families. And what we found is actually neuroligin 2 could be ubiquitinized by each, and this is when we put different each family members together with neuroligin, and we immunoprecipitate down and Western blot with ubiquitin. You can see that each could ubiquitinize neuroligin 2. And among those family members, there's two more family members that, especially the NEDD4, can also ubiquitinize neuroligin 2. So, and as well as HECW2. So this raises the question whether this ubiquitination of neuroligin by each is any specific, or there's other protein E3 ligase might also involved in the ubiquitination of neuroligin 2. And when we look at the possible function, we know that many of the ubiquitinized protein could undergo endocytosis. So we look at the endocytosis of neuroligin 2 under together with all the each family member. What we found interesting is that only each was able to induce endocytosis of neuroligin 2, as shown in here. That so this is the vector control. You can see that neuroligin 2 is expressed on the cell surface, and when we express together with each, you can see majority of neuroligin 2 actually will be endocytosed in the intracellular. However, when we co-express with all the other each family member E3 ligase I mentioned earlier, including NEDD4, which can also heavily ubiquitinize neuroligin 2, we don't see any surface change. So when we did another assay, which is the biotinylation assay, look at this quantify the surface expression neuroligin 2, we find a similar result, which is that only each could significantly reduce the surface expression of neuroligin 2, while NEDD4 as well as HAC1, HACW2 doesn't do anything on the surface neuroligin 2. So this actually did indicate to us that there's some specificity, although two other E3 ligands could also ubiquitinate neuroligin 2, but this ubiquitination itself seems to be not sufficient. But however, each not only can ubiquitinate neuroligin 2, but can also induce the surface reduction of neuroligin 2. And we look at what could explain this specificity, and it turns out that not only that we need the ubiquitination of neuroligin 2, we also need the interaction. In other words, each not only need to ubiquitinate neuroligin 2, but also need the binding capability. In addition, together, they can bring down the neuroligin 2 from the cell surface. And this will generate a number of, we mapped the region that responds for the interaction. Now it turns out that it's a WW domain that is responsible for interaction between each and neuroligin 2. And when we delete this domain, and we will see that when we delete this domain, that this is called no 1, 2, 3 WW. This could no longer reduce the surface expression of neuroligin 2. Well, as long as we have the interaction domain and also the ubiquitination domains, we'll be able to do this. And when we did the cell staining assay, we see the same result, that without this interaction domain, we won't be able to have surface reduction. And without, of course, without the HAC domain, which is a ubiquitin ligase activity domain, we will not see any reduction on the surface as well. Well, as long as we can bind and we can ubiquitinate neuroligin 2, and we will be able to bring the neuroligin 2 into intracellular site and reduce the surface expression. So this requires the simultaneous binding and ubiquitination is required for reduced neuroligin 2 at the surface cell surface. And we also look at what could this ubiquitination and also surface reduction of neuroligin 2 could do to the synapse formation. And we look at we expressed each, as well as we generate a mutant of each called each C830A, which is a mutant that will eliminate the ubiquitin ligase activity of each. What we found is that if we express the Y-type each, it will significantly reduce both the Jeffrin as well as gamma-2 at the inhibitory synapse. In other words, Y-type each will reduce the inhibitory synapse formation, while when we mutate the the E3 ligase activity, it won't lose capability to do so. And we also knock down each in the neuron. What we found is that when we knock down each, we see the opposite effect. We see actually the increase of the inhibitory synapse number and neuroligin 2 in terms of neuroligin 2. And also when we look at the inhibitory miniature potential, we also see when we knock down the each, we see an increase of miniature iPSC, which is indicating that the inhibitor synapse number has been increased. Okay, this is pretty much what I want to say today, because I was told that I should keep it short so we can have more time for discussion. So I want to thank the people who have done the job. So the job I described today is mainly done by two talented graduate students, which is Honglei Tian and Chen Tan, as well as a collaboration with Zhejiang University Jianhong Law School and Jun Xia, Yong'an Du, Zheng Rui, and Paiding Liao for the electrophysiology studies. I would like to thank you all for the time, and I think, okay, yeah, one more thing. So actually, as Zilong just mentioned that I've been working in HKUST, which is Hong Kong University of Science and Technology for 20 years now. But in the coming months, actually next month, I'm moving to Guangzhou because HKUST just established a new campus in Guangzhou, and it's supposed to be much more grand than the campus we have in Hong Kong. And you are welcome, all of you are welcome for a visit when time and the situation allows. Thank you all for the time.
Thanks, Jing. That's a beautiful campus they've been. So now to start our questions, I might start with first question. So for the each protein is very interesting. So is that self-inhibition effect or some activity dependent repression you can imagine to repress the inhibition synapse formation?
Yeah, that's actually a great question. What I can say is yes, we can observe that ubiquitination of neuroligin is associated with neuronal network activities, and that can also regulate the synapse formation. But at this moment, we are still working on to pinpoint whether each is specifically play a role in this diagram.
Okay, so as I imagine, so ubiquitination will be a transient process, right? It will not be attached all along, because for the protein omega acid, you have a very strong. There has to be something like attached on the neuroligin 2 intensively, at least.
Yes. You're absolutely right. It's actually a dynamic process. And actually, in addition to each, which is the E3 ubiquitin ligase, we actually also identified a D-ubiquitin ligase.
Okay.
So it's, yes, you're right. It's a dynamic process.
Great.
We have identified about a dozen proteins from that immunoprecipitation, and we're still trying to figure out all the puzzle together, how to put them together.
Great, thanks. Okay, any more questions?
Hi, Jun. This is excellent, really nice, very exciting data. So I was wondering, how does Jeffrin fit into this?
Oh, okay. Very good question. So we were able to show that alpha could bind with both neuroligin as well as bind with double receptor, which is a Susumo's group original discovered. But when we try to figure out whether it's linked to Jeffrin, we still did not find a direct interactions. So you can get Jeffrin in the picture. We can see Jeffrin gets on with alpha, and when we knock down alpha, Jeffrin goes away. But when we try to find the direct induction, we still couldn't. So there might be other partners that link either alpha or neuroligin or GABA with Jeffrin, but we have not found that particular linker yet.
If there's no more other questions, I can maybe expand this a little bit more. There in C. elegans, work from Josh Kaplan's lab has identified this FRM3, which is a protein 4.1 Ezrin Moisin family of proteins, and that seems to also be important in generating this pool of GABA receptors that are very immobilized at the synapses. So I wonder, in your proteomics hits, whether you've seen these cytoskeleton like proteins that are directly interacting with the receptor.
Yeah, that's a very good question. So we did identify cytoskeleton protein, but those cytoskeleton proteins are very sticky. In other words, they also come down from the knockout mice controls. Now we have not actually getting to the stage to quantify the interaction yet. So right now we are focusing on the protein. We know that it's clean, which means we did not identify anything from the knockout mice. But I know that some of the protein we can see some difference, but we have not got to the stage to quantify them. I would imagine some of the cytoskeleton protein might be involved, but we have not got there yet. You're right.
Very cool. Thank you.
Thanks.
I think Jing Zhao, do you want to ask a question yourself? Your question on the chat bar. Yeah, welcome to unmute yourself and ask Jing directly.
Sorry.
Yeah, go ahead. Go ahead.
Can you hear me? Yeah. Hi, Jing. Actually, I have a question. I find your first slides you showed in the LHFPL4 knockout mouse. They have colocalized with the very good colocalized with the neuroligin 2. So I just want to know whether the inhibitory static transmission in the knockout L4 knockout mouse have a decrease or didn't have any change.
Oh, okay. And that's a good question. We actually did email scanning, which we found that in certain brain region, so not all region actually, especially in the cerebellum, the inhibitory synapse number are significantly reduced, and the mice are more likely to have a seizure. But one thing I probably did not say is that it's called L4. There's actually a number of family members. You have L1, L2, L3, L5, L4, L5. So my guess is that if we look at the whole brain, some of the brain region is being compensated. In certain region, we can see a significant reduction of inhibited synapse. Yes.
Also, I have another question. Sorry.
Yeah.
I want to know the L4, the protein, whether can co-localize with the neuroligin 1. You said the neuroligin 1 and the neuroligin 2 have very similar sequence. So.
No, it doesn't not co-localize with neuroligin 1 because neuroligin 1 is mainly in the excitatory synapse, and that we are certain about it. But for neuroligin 3 can be show up in both inhibitory and excitatory synapse. It does have some co-localization with L4.
Okay, thank you.
Okay. Maybe I have one last question for the super resolution data. I love the data so much. So currently, we also have some candidates synaptic protein, which is called a CNTNAP3, which is, but we have no idea what is it pre or post synaptic. So would you have, Jun, so what's your suggestion? So should we also have some, that was our culture neuron, right? And the super resolution of the pre or post.
That super resolution, I like that picture also.
Yeah,
one reason is because that's one of actually we got this super resolution before the commercial super resolution was microscope was ready. It's actually the homemade. Yes, you can just use GAT and Jeffrin to do co-staining with whatever protein you like, but you have to do it separately. We were not able to achieve 3 color. We have to do 2 colors separately with super resolution.
Yeah, I'm curious about the culture neuron. Is that a clean cut, or is also be, or sometimes there will be a majority of the will be the mixed with Gafferin, or sometimes it will be pre-synaptic, or you see the post-synaptic all the time?
Oh, this is quite clear cut. No, this is you can see this is the zoom out, zoom in picture, but you can see the big one. You can see here. You can see those are color quite well. But in here, you can see clearly separated.
Okay, great. Thanks.
You don't need to quantify. Just look at it under your eye. You will be able to tell.
Amazing. But,
the key problem is you need a good antibody. Super resolution. You really need a very good antibody. Cat 65 and Jeffrin are good markers. Then you need to have your own antibody that are as sharp as those antibodies.
I will ask you more for the detail for the where antibody from. Thanks.
No problem.
Okay, so any more questions, Aaron? Should we do the second talk?
Great.
Thanks, Jane.
Okay.
No problem. Nice to see you guys.
Thank you.
Thank you very much, Jun. Great talk. And now it's my pleasure to introduce the next speaker today, Dr. Ching Su. Ching is an associate professor in the section of neurobiology, the Division of Biological Sciences at UC San Diego. She earned her bachelor's degree from National Taiwan University and then did a PhD in neuroscience at Johns Hopkins. And here she worked with Qingwei Yao. And she had this amazing paper in science. Actually, she's had 2. Well, anyway, first things first, she had this amazing paper in science as a graduate student where she studied lizards and frogs and salamanders and things like that. Some sharks have a third eye called a parietal eye, and that's used for like telling if it's like sunrise or sunset, not like making images. And they have in that eye photoreceptors that are kind of like rods and cones, but they have this feature that other photoreceptors like our eyes don't have, in that different lights, like a blue light can cause a depolarizing response, and blue light can cause a hyperpolarizing response, and green light can cause a depolarizing light response. The mechanism of that was unknown. She cloned both of those components, the opsins that mediate this, worked out the signaling pathway. It's really an amazing paper. And then she continued working on sensory systems as a postdoctoral fellow at Yale with John Carlson, here turning to Drosophila. And here she worked on this phenomenon that in sensory systems, there are the sensory cells are usually grouped together, like taste buds and stuff like that. And there needs to be a way of distinguishing different sorts of stimuli, different odors, different tastes, and things like that. And how that's done is not known. She focused on olfactory receptor neurons in Drosophila, and she figured out that there's this sort of lateral inhibition signaling that does not require synapses. It's called ephaptic coupling. So that actually I never heard of that word before. So and then there's another word that I had never heard before, in that she worked out the way this works, and it's called valence opponency, and that's in the title of her talk now. She'll provide more insight into it, but it's a really elegant mechanism in which different stimuli can be integrated into a behavior, and she'll tell us more about that work now. So thanks, Chih-Ying. Please take it away.
Oh, thank you so much, Aaron, for the kind introduction and also for the opportunity to share our research with you today. So my lab studies the sense of smell, as Aaron mentioned, that using Drosophila as a model organism. And so the basic olfactory circuit structure between mammals and insects is conserved. In both systems, olfactory receptor neurons that express the same receptor project an axon to the same glomerulus with the synapse onto second order neuron to further carry information to higher brain region. And the majority of the olfactory receptor neurons is considered as a generalist because these neurons can each respond to a wide range of odorants to different degree. And as such, each odorant can activate a unique combination of receptor neurons, and this activation pattern can in turn encode the identity of the odorant. And by extension, this activation pattern likely also defines the hedonic value of the odorant, that is, whether it smells attractive or aversive. And therefore, olfaction is believed to employ a combinatorial code with each neuronal type operating as the equally weighted input channel. And similar to the English alphabet, which by itself does not have any specific meaning, but when put together in unique combinations, they can then support a large vocabulary to encode a vast number of odors. But on the other hand, a small minority of the receptor neuron is considered as specialist because neurons apparently only respond to one or two ethologically meaningful odors, such as the pheromones or predator odors, to mediate robust innate behaviors, such as courtship, aggression, or escape behavior. Therefore, the specialist neurons are considered to follow a label line code, rather like the emojis. And the functional purpose of the primary sensory neurons is thought to produce a symbolic representation of the stimulus. But on the other hand, information processing is believed to occur only in the central circuit. However, we think this division of labor perspective may be too simplistic to fully represent the olfactory circuit function, especially for insects. So our inspiration comes from this phenomenon of sensory neuron compartmentalization. So across animal species, many primary sensory neurons are compartmentalized within specialized structures, such as the taste buds on our tongue, or a variety of invertebrate sensory hair known as sensillum. So intriguingly, compartmentalized sensory neurons typically respond to unique combinations of stimuli. For example, the pairing of sugar and bitter sensing cells in our taste bud, or in the insect gustatory sensillum, or the pairing of moist and dry sensing neurons in the hypersensory sensillum, or the cold and warm sensing neurons in the thermosensory sensillum. So within each compartment, we wonder whether the close proximity between sensory neurons can somehow allow those neurons to directly communicate with each other so as to provide a means to pre-process sensory inputs before they reach the brain. So to test the possibility, we use a fly olfaction as a model to determine whether compartmentalization has any functional impact on how those neurons respond to odorants. So fly olfactory sensillum can be found on the surface of the antenna. So to study the function of these neurons, we perform extracellular single unit recording to monitor their spike activity. So for most sensillum types, the spike activity of the group neurons can be distinguished based on their characteristic spike amplitudes. So for convenience, we name the neuron with the larger spike amplitude, the A neuron, and the smaller one, the B neuron. So in an antenna, there are about 50 different types of olfactory receptor neurons, which are grouped usually in 2 and up to 4, in a total of 20 different types of sensillum. And strikingly, the grouping of these neurons is genetically predetermined. For example, in the AB3 sensillum, the large spike A neuron expresses the 22A receptor and is always paired with a small spike neighbor that expresses the 85B receptor. So the question is, so what is the functional meaning of this highly organized stereotype grouping of olfactory receptor neuron? And in addition, does this difference in spike amplitude indicate any functional distinction between the A and the B neurons? So to address the first question, we recorded from the AB3 sensillum. So first, we presented an A neuron specific odorant in the background to chronically activate these neurons. And next, we apply a pulse of a B neuron specific odorant to transiently activate the B neuron. And interestingly, we noticed that strong activation of B neuron was also accompanied by the inhibition of the A neuron response, as shown here in the sample trace or in the average spike response over time. So this result raised the possibility that strong activation of the B neuron cross-inhibits its neighbor. So to test this hypothesis, we genetically ablated the B neuron by expressing a cell death gene reaper specific in those neurons. So if the A neuron response was inhibited by B neuron activation, then killing this B neuron should also abolish this inhibition, right? And we found that was indeed the case, normal inhibition of the A neuron by the B neuron odorant. So taken together, this result indicates that compartmentalized olfactory receptor neurons can indeed functionally interact with each other, in that the strong activation of the B neuron can inhibit the response of the A neuron. So without showing you the data, we found that strong activation of the A neuron can also likewise inhibit this neighbor. So in other words, this cross inhibition is bidirectional. But how about the mechanism which underlies lateral inhibition in the sensillum? So much to our surprise, we found such inhibition actually does not require any chemical synapses. So shown in this sample trace, we could still observe robust lateral inhibition even after we block synaptic transmission by expressing tetanus toxin in both neurons. So if it is not through inhibitory synapses, which other mechanism could mediate lateral inhibition in the sensillum? So we think effective coupling is likely the answer. So the term effective coupling refers to direct electrical interactions that arise from the close opposition of neuronal processes from two or few neurons. And this mechanism looks really promising because the sensorial environment seems ideal for effective interaction to occur between neighboring neurons, given that their sensory dendrites are in close proximity of each other in a very well-insulated microenvironment. The question is that what kind of experiment can we do to test this hypothesis if the interaction is enabled by the microenvironment instead of some proteins or ion channels that we can manipulate? So to address this question, my former graduate student Ye performed a really cool experiment. So instead of recording from a single sensor with a glass electrode, Ye used a tungsten electrode to record two nearby sensors at the same time, such that this metal electrode can connect the electric field of these two sensors. So if the electrical interaction is sufficient to mediate lateral inhibition between olfactory receptor neurons, then in this configuration, strong activation of the orange neuron is then expected to inhibit the response of the blue neurons. And we were so excited to find out that was indeed the case. Here we can see the concurrent excitation of the orange neuron and the inhibition of the blue neurons. And as a control, we withdrew the electrode from one synapse such that the orange and the blue neurons were no longer electrically coupled. And as expected, this decoupling also abolished the inhibition. So taken together, this result indicates that electrical interaction or effective coupling is sufficient to mediate electrical inhibition between electrically coupled olfactory receptor neurons, so which is normally the case between neurons housed in the same synapse. So without showing you data, Ye further found that the large spike A neuron can actually exert greater effective influence onto a small spike neighbor than the other way around. In other words, for compartmentalized neurons, the difference in their spike amplitudes actually indicate functional differences between the A and the B neuron. But what could possibly underlie this functional asymmetry? So given that larger neurons will have smaller input resistance, which can in principle allow these neurons to have greater effective influence, we think this functional asymmetry likely arises from a scenario where the large spike A neuron is physically larger than its neighbor. So to test this hypothesis, my former student Angela teamed up with Eric Bouchon at the National Center for Microscopy and Image Research at UCSD to first develop a tissue preparation method which allows us to perform high quality morphological preservation of genetically labeled neurons for volume electron microscopy. So Angela further generated transgenic flies in which a genetic encoded EM marker named APEX2 is selectively expressed in one of the group neurons. So APEX2 is a peroxidase which labels neuron by converting diamethylbenzidine into electron-dense polymer visible under EM. Then we then image the antenna with serial blood phase scanning electron microscopy. So in an automated process, this technique removes a thin layer of tissue before the region of interest is imaged. And this sequence is repeated hundreds of times until the entire volume of interest is imaged for 3D reconstruction. So here are some simple images taken from a pair of AB3 neurons where the A neuron expresses APEX2. So you can see the darker labeling here. So using these images, we were able to construct 3D model for the labeled A neuron, shown here in blue, as well as its neighbor shown here in orange. And it was immediately obvious to us that the large spike A neuron is indeed larger than its neighbor. So in addition to the AB3 synapse, we found that the neuronal size difference was broadly observed across synapse types. And here is my favorite example. So in this AB2 synapse, reconstructed by my students Cesar and Quentin, we can easily appreciate that the large spike A neuron, shown here in blue, not only has a larger soma than the B neuron, but also has far more dendritic branches than its neighbor, as shown here in this EM image or in the 2D projection. So this one has over 300 dendritic branches and the neighbor only has one. And so with this wealth of morphometric information, we are currently investigating how these striking differences in dendritic morphology and sensory surface area can influence olfactory function. So to summarize the first part of my talk, we found that compartmentalization enables fly olfactory receptor neurons to directly communicate with each other through effective interaction. And interestingly, the strength of this interaction is asymmetric in that the largest spike A neuron are dominant. And mechanistically, this functional asymmetry arises elicits in part from the size differences from compartmentalized neurons. So in light of this specific neuronal interaction, we should see each synapse type as an olfactory processing unit comprised of functionally distinctive input channels. And in sensory neurobiology, electron inhibition is best known to enhance the contrast between parallel sensory inputs. So in this context, we are very curious to know what kind of information is then compared and contrasted by lateral inhibition in the sensorium. So to address this question, we first surveyed published literature in which many flight olfactory receptor neurons have been individually characterized. For example, in the AB1 sensorium, that there are 4 neurons, and 2 of them have large spikes and 2 have small spikes. And it has been shown that both of the large spike neurons mediate flight attraction to vinegar, and one of the small spike neuron mediate flight aversion to CO2. And interestingly, many positive balance behaviors, such as attraction, enhancing egg laying, or promoting courtship, are associated with large spike neurons. And in contrast, the characterized small spike neurons instead mediate negative balance behaviors. And as I mentioned earlier, AB1's large spike neurons mediate attraction, while one of the small spike neuron mediate aversion. And in the AB4 sensorium, the A neuron enhances egg laying, while the small spike neighbor inhibit the same behavior. So based on these observations, we hypothesized that compartmentalized flight olfactory receptor neurons can antagonistically regulate the same type of behavior, with the large spike neuron promoting the behavior while the small spike neighbor inhibiting it. So we know that this published papers focus on 3 different behavioral contexts: place preference, egg laying, and courtship. So to keep our study within the framework of these findings, which we consider to be the ground truth, we decided to test our hypothesis by also focusing on these 3 behavioral contexts. Unfortunately, the receptors of these neurons have only been identified, shown here in the parentheses. So this gives us a genetic handle to selectively target each neuronal type. So this project was led by my graduate student, Ching Su, who worked closely with the master student, Vanessa, and the former postdoc, Jay. So to selectively activate the target neurons, we use a receptor-specific Galpo drivers to express the red-shifted channel rhodopsin called CS crimson, or the warmth-activated cation channel TRPA1 in either the A or the B neurons. And we then examine whether red light or warm temperature can bias this flight behavior response in one of the 3 contexts. So for example, in 84 sensorium, the large spike A neuron can promote male courtship in response to a pheromone. However, whether the BOC neuron had any role in courtship regulation was unknown. So to address this question, Ching Su expressed TRPA1 in one of the 3 84 neurons, such that the target neuron can then be activated by raising the temperature. So as a positive control, he first focused on the A neuron in the courtship assay, in which the experimental male fly compete with 2 parental control male fly to mate with a wild-type female at 29 degrees. And so here, a plus of cumulative copulation rate over time. And we can see that the males whose 84A neurons were thermogenetically activated, shown here in blue, held a copulation advantage over the control flies. So this result successfully recapitulated the courtship promoting role of 84A and thereby validated our thermogenetic active courtship assay. And using this assay, Ching Su further found that both of the small spike 84 neurons can instead inhibit courtship when thermogenetically activated. So taken together, these results support our balanced opponency model in the context of courtship regulation. So in the interest of time, I'm just going to show you a summary of our results. So we have examined all 23 neuronal types. So this is in this table. And we found that the majority of these neurons have behavioral output that follow the corresponding balance opponency, suggesting that compartmentalized olfactory receptor neurons are organized in the sensorium based on their opposing balance. So in summary, we found that balance opponency is broadly observed between compartmentalized olfactory receptor neurons across the sensorium type in multiple behavioral contexts. Therefore, we can now view each sensorium type as olfactory processing unit for balance information. And this organization likely provides a peripheral mechanism for preprocessing countervailing olfactory input by letter inhibition and synthylum, so as to facilitate appropriate behavior response for the animals. So in light of these findings, I would like to revisit the coding principle for olfaction. We now think that the dichotomy between specialists and generalists is likely too simplistic a view on olfaction. For lizard fruit flies, we found that the majority of the olfactory receptor neurons carry some intrinsic valence value to inform important innate behaviors, regardless whether these neurons respond to a large or small number of odors. However, built on this valence structure, we think these neurons can still generate a combinatorial code to represent different odor identities. So in this sense, each olfactory input channel is probably more similar to a Chinese characters, in that each character carries some intrinsic meaning, but when put together in unique combinations, they can then support a large vocabulary to encode information which can transcend the character's original meaning. So with that, I'd like to thank my current and past lab members for making this study possible. I'd also like to thank my funding sources and collaborators. So thank you very much for your attention. I'll be happy to take questions.
Great, thanks, Chih-Ying. Now we're open for questions. I might start with one question. So have you ever seen effects on if you have more than one odor that activates the same, or that activates different odorant receptors, and then the effect on behavior?
So to co-activate both neurons at the same time, is that?
Yeah.
Yes. So we didn't show it here, but actually we can demonstrate that effective interaction can modulate behavior, even without passing on the synaptic information to the brain. Yeah. So we can actually block synaptic transmission for one olfactory input channel with a tetanus toxin, but then while maintaining the effective interaction in the periphery. But in that context, we can still see an impact on behavior. Yes.
Got it. Okay.
Hi.
Could I ask a question?
Yes.
I'm looking at this beautiful EM reconstruction and the branches you're finding. And presumably there will be GPCR receptors in individual, the huge branches.
So the receptors are ligand-gated cation channels. Yes. And we don't know whether they are GPCRs on the sensory dendrite or not. Yes.
And do you always see this large body size correlating with expanded dendrites?
Yes, we actually see a correlation between the spike amplitude with dendritic branches. So the more branches there are for that neuron, the larger the spike amplitude is. Yeah. So, and it's really striking to see that the olfactory receptor neuron can have such a diverse morphology, because I think in the past people believe that although there are a lot of neuronal type, but the only difference is the receptor they express, right? And people actually did not expect that each neuronal type actually have a different function and different morphology.
And oh yeah, there's another question someone else can ask.
Kai-Wen.
Hi, Ching. Great talk, very interesting. So I'm just curious about how common is this emphatic coupling exist in the brains? Because in the olfactory systems, so you notice that there's the large and small cell body, the soma size determine the couplings, right? But in many of the brain regions, the large and small cell body are very commonly observed. So I'm just curious how common is that in the brain across different brain regions. And also another question is, how does the emphatic coupling regulate it? Is that all or none, happening in an all or none manner, or can be like the strengths of the coupling can be modulated in some way?
That's a great question. So to answer your first question, how common is this? The short answer is that it's actually quite common, but difficult to study. So it was reported in the fish, tailless fish, that the difference, but there's effective interaction between motor cell and the interneurons. And the motor cell is relatively large, and the interneuron is quite small. And this also happens in the cerebellum in all vertebrate system, and between the Purkinje cell and basket cells. And so the environment that's conducive to effective coupling is that you need closer position of neuronal processes, but you also need to have an external environment that has high resistivity. So like current can flow from one neuron to the other, but not to the environment. So the short answer is very common, but hard to study. Right, because you have to maintain that environment in order for effective coupling to exist. And the second question that is the strength of effective interaction modulated? Then the short answer is also yes, it's activity dependent, right? So even though the large spike neuron, the larger neuron can have a greater influence onto its neighbor, but this interaction, the degree of inhibition is activity dependent. So typically the neuron that has a higher activity can have a greater inhibitory effect onto its neighbor. So the activity dependent.
I see. So the emphatic coupling is always delivering the inhibitory effects, but not excitatory effects.
That's an excellent question. No. So the inhibition is only observed when both neurons have a high activity. So it's non-linear. Yeah. So we often wonder that what's the benefit of using effective interaction instead of synaptic interaction? And so one benefit is that it's kind of metabolically more, it's much cheaper. It's not as expensive as synaptic transmission, but it's also very fast. And also there's activity dependent. So there's non-linearity in this as well.
Okay, very interesting. Yeah, I hope that in the mammalian systems we can adapt the way that you used to study the emphatic couplings. So yeah, thank you.
The flight system is much user friendly, I have to say. Yes.
Yes, thank you.
Okay.
More questions, Jin? No. Okay. Anyone else? No. Okay. Awesome talk, Ching. Thanks so much.
Thank you very much. See you at the conference.
Yeah, see you. Yeah, see you, Zilong. Thanks so much.
See you.
Have a good time at your conference.
Enjoy Ventura.
Yeah, we miss you, Zilong.
Yeah.
All right, see everyone. No Neuro Zoom next week, but then